Hello everyone, welcome to Supplement Tutorials. Today we'll be talking about liver function test. And before you watch this video, I would like you to hit on the subscribe button. Alright, so let's keep going. Okay, so when we talk about liver function test, what comes to mind? We are talking about liver, we are talking about the function of the liver. So liver function test is a biochemical test that is used to assess the function of the liver. And someone might have a liver problem, someone might have a hemolytic problem, anemia problem, or someone might have some kind of issue with the liver and want to find out what is actually causing the damage, or what is actually causing this particular signs and symptoms like jaundice, etc., etc. Well, the only test that will be used to diagnose the liver infarction or the liver disease is the liver function test. Alright, so how do we categorize liver function test? Well, basically, liver function test is grouped into three major categories. First is the part of the test that assesses the synthesis ability of the liver. The second is the part of the test that checks the rates at which the liver excretes certain substances, and then finally it checks the damage of the liver. So let's talk about the synthesis aspect of the liver function test. So the liver actually synthesizes proteins. One of the major plasma proteins the liver synthesizes is the albumin. Albumin is responsible for about 60% of the total plasma protein that we have. Aside the albumin to the other proteins like the globulins. Alright, so it's the liver that synthesizes these proteins. And if there is a decline in albumin, or if there's a decline in globulins, or if there's a decline in the total protein, then we can say that there's something wrong with the liver that is reducing the production of what? Of this protein. And if there is also excess production of this protein, like hyperalbuminemia, that is excess production of albumin, or there is excess production of the globulins, then we can say that there is something wrong with the cells, with the liver cells, that is causing the excess production of what? Of these proteins. Probably it could be a hyperplasia or something. Alright, now, so what happens when the total protein or the albumin drops? So when albumin drops, there can be symptoms like edema. This is because albumin is responsible for the control of fluid balance within the intravascular compartment and then the interstitial space. So albumin exerts a pressure called the oncotic pressure, and then if there is a decline in the albumin, the oncotic pressure reduces. So when the oncotic pressure reduces, there's a pressure called hydrostatic pressure. This hydrostatic pressure moves fluid from the intravascular compartment or from the blood vessel into the interstitial space, whereas the oncotic pressure keeps the fluid within the interstitial space, within the intravascular compartment. So when there's a drop in the oncotic pressure, hydrostatic pressure will increase. That will drain fluid from the intravascular space into the interstitial compartment, and then it will eventually fill up the lymphatic nodes or the lymphatic vessels, and then to cause what we call edema. Alright, now let's go to the excretory ability of the liver. Now the liver, as we know, is responsible for bilirubin excretion or bilirubin conjugation. Now after red blood cells have broken down in the spleen, they are carried to the liver to undergo what we call bilirubin conjugation. That is how bilirubin synthesis is actually done. So after the red blood cells are broken down, and then they eventually end up to become what? Unconjugated bilirubin, and in the liver, that is where they are conjugated and then excreted in the bowel. Alright, so if there is in the excretion aspect, there are three parameters. We have the total bilirubin, we have the conjugated bilirubin, and we have the unconjugated bilirubin. So the total bilirubin is a combination of the conjugated bilirubin and the unconjugated bilirubin. And the unconjugated bilirubin actually has to be conjugated. And the conjugated bilirubin has to be what? Excreted in the urine or in the stool. But then when we find, conjugated bilirubin or unconjugated bilirubin in the systemic circulation, abnormally in the systemic circulation, that means there is something wrong with either the conjugation of the unconjugated bilirubin in the liver, or the excretion of the conjugated bilirubin into the bile duct. Yeah, so that is what this particular parameter assesses. Now, in units, it is essential for us to conduct liver function tests for them to assess their serum bilirubin levels. Now, when we realize that the baby's or the neonate serum bilirubin levels are high, then it means the baby is at threat of having a condition called kinecteros. Now, unconjugated bilirubin is lipid soluble, and it is able to cross the blood-brain barrier to cause neurotoxicity or a condition we call kinecteros. Now, in order for us to prevent this one, we have to allow the baby to undergo a therapy called phototherapy. This phototherapy converts the unconjugated bilirubin to photobilirubin, which is much excretable in the body. So when there is excess unconjugated bilirubin, or when there's excess bilirubin in the bloodstream, then it means it's likely to cause what conditions such as jaundice and kinecteros. That's in units. And also albumin is also responsible for the transport of these unconjugated bilirubin to the liver for it to be conjugated. So when there is a drop or a decline in albumin, such as hypoalbuminemia, it means there will be a lot of unbound unconjugated bilirubin, and these unbound unconjugated bilirubin can lure themselves into the bloodstream, and then find its way to the brain, to cross the brain barrier and cause kinecteros. But it's really rare in adults and mostly common in infants. All right. So finally, let's talk about the destructive ability of the liver. Now, in the destructive ability, or in the tests, in the part of the LFT or the liver function test that assesses how the liver has been destroyed, it is an enzymatic assay. In the hepatocytes, there are certain enzymes that are found that are responsible for biochemical synthesis and biochemical reactions. These enzymes include, we have ALT, alanine transaminase or transferase. We have AST, that is amino aspartate transferase. We have GGT, that is gamma glutameral transferase, that's GGT. Then we have I think ALP, alkaline phosphatase. Alright, so these enzymes are actually found within the hepatocytes, and when there is cellular damage, whether it is apoptosis, whether it is necrosis, these enzymes are released into the bloodstream and are detected. So for us to know whether the liver has gotten destroyed or the liver has damaged, we have to actually assess or look out for these enzymes. Now, ALT is liver specific. Whenever there is an increase in plasma ALT, then it tells us that there's something wrong with the liver. It could be viral hepatitis, it could be cirrhosis of the liver. And then moreover, AST2 is also found within the cells of the liver. And when you find AST circulating in the plasma in a maximum quantity, then it can tell that there's something wrong with the liver. But then AST is also found in cardiac muscles and some other tissues. So therefore, you can't really tell if AST, the cause is really of a liver or from another source. So in order for you to distinguish between a liver disease or a non-liver disease based on AST, there's something we call AST to ALT ratio. Now we compare the ALT, the AST to the ALT. The ALT is liver specific. So when we realize that AST to ALT ratio is less than 1, then we can say that the particular liver disease is an alcoholic liver disease. When we realize that it's greater than 1, it is known as non-alcoholic liver disease, and it could be as a result of viral hepatitis or cirrhosis. Now, when we realize that AST-ALT ratio is greater than 2, then it's an indication of severe alcoholic liver disease. This means that AST has increased and ALT2 has increased. Therefore, it is as a result of severe alcoholic diseases. Alright, so aside AST and ALT, ALP2 is found in the lining of the bile duct, and it is mostly seen in the bloodstream when there are conditions such as hepatic jaundice or obstructive jaundice. That is when we can find this ALP in the bloodstream. And also ALP is also found in bone tissues, and when there is an increase in ALP, plasma ALP, we can say that it's as a result of overproduction of the bone cells or something that has caused an increase in the ALP. And also in leukemias to ALP can also show in the plasma. Alright, now let's talk about the last enzyme, that is the GGT. So GGT can also be seen in obstructive jaundice, but most importantly, GGT is used to assess alcoholic condition. When you take a lot of alcohol, GGT can elevate, and mostly GGT is elevated in in males than in females.